Good, you all here today. I'm going to share my screen and I'm going to actually turn my video off because I had some bandwidth issues, but good to be with you all. Good to be sharing the stage with Josh, and we are talking about fava bean disease management today. And I guess beans have gotten my interest recently. I'm keen on the rotational benefits they can provide, the nitrogen they fix, the options they give in double breaks with canola. There's a lot to be excited about, but we do need to get the yield stability better. And one thing that's holding us back is that disease is a consistent risk for us in the high rainfall zone. There are years where it's not as bad as others, but it's consistent. It's always there, always got to be thinking about. And managing it, I think people are feeling more and more confident about looking after their beans, but it's still a significant variable cost. So the question is, what are the options? How do we put them together? These are things that we're always thinking about, so it's always good to still keep chipping away at it in our research program. So what are the options and how they put them together brings me to Josh. Over to you, integrated disease management.
So the most important thing about disease management is an integrated approach to it. And so on the screen is listed a few things that basically we're going to go through today, and we're going to go through some data from some fantastic trials through the HRZ. So fantastic that we're worried about disease overrunning everything, even our complete controls, because we just can't get enough fungicide out there. And that's the problem with susceptible varieties, and they actually impede on our more resistant varieties as well. So that brings me to the first point. It's about growing a more resistant variety, ensuring you know what's around and growing the best variety that you can. And at the moment, as Craig has talked about, and Scott, PBA Amberley is probably our best variety in the HRZ at the moment for disease. It is a step above everything else, but by no means a miracle bullet that we thought it might be based on the original data or original conversations. And we'll go through that in a minute. It's about good paddock rotations. So if you've got good rotations, you haven't had the same crop or disease in that paddock over the last 3 to 4 years, you're not going to have that inoculum in the paddock already that's going to infect your next crop. It's understanding the seasonal effect on disease. And we're going to go through season today because there's quite stark differences between Lake Bolac and the Lake Linlithgow sites. And we've actually dropped out all the other sites that we've got. These trials, that some of these trials we're looking at and going through results are actually replicated across the state in 5 or 6 locations. And the data through the HRZ is just a lot better, and there's a lot more disease incidence, because in those other areas where there isn't as much rainfall or the environment hasn't been as conducive to disease, we haven't seen the disease develop. The next point is fungicides should be our last resort. So use the resistance where we can, use good agronomy, good paddock rotations, and then rely on our fungicides as that last resort and to mitigate that disease during the season. And it's important to rotate between those. We're losing fungicide actives in cereals, and this could go the same way in pulses. We've got a significant concern with it's a fantastic time to be in pulses because there is so many fungicides being registered. All the chemical companies are bringing across everything from cereals to pulses. They're highly effective, and we'll look at these after. But it's about ensuring that you rotate between these actives. Don't use them in your cereals, then your canola, then go into your pulses with that same fungicide active 3 years in a row, because we're just going to break them. That's why we need to use an integrated disease management approach. Now, a few other points there that I feel fit into an integrated disease management approach, but it's just about general planning. It's have a plan in place, monitor for disease, so know what's in your crop, know what diseases are in your area, and then know where and when to get help. We don't know everything. I'm the first one to say that I don't know everything. Been in this area for a few years, 7 years in agriculture Victoria and research. I'm still learning a lot, and I'd be hard pressed to say anyone that's talking today wouldn't keep learning on what we're doing. Next slide, James. We're just going to run through some of the diseases now, very quickly, just as a recap. Cercospora leaf spot starts off very early in the season, pictured there, usually runs out of legs as temperature warms up. It can get quite aggressive, and there's no resistance to Cercospora leaf spot. So all our varieties are basically the same. Next slide, James. Ascochyta blight. This will come in at the same time as cercospora leaf spot. We'll keep going through the season, but usually our chemical controls that we use for some of the other diseases usually hold off ascochyta blight. It can get aggressive at different times through the season, and it's just about monitoring. Next slide. But the biggest threat that we're seeing, and the most aggressive disease that we see, is really chocolate spot, and it can go from 0 to 100% leaf area infected in a short period of time under really conducive conditions. But the really big trouble is that it's really, the flowers are susceptible, and so as soon as it starts affecting those flowers, they'll start dropping off. And we can see this in the sites this year, that as soon as those flowers are getting infected, they drop off and you've basically lost your pods. So you're losing that grain yield immediately from that. The other thing is the pods are quite susceptible. So as soon as your chocolate spot gets on your pods, you're then starting to get your seed staining, and pulses are all about that seed quality, unless they're going down sheep's throat, I suppose. And the sheep aren't too phased about that. But you're still going to lose your grain yield, and it can still affect that quality for that marketability. So on to some trial results, or disease ratings first, I think. So I think I was going to recap quickly about some disease ratings. So cercospora leaf spot, everything's susceptible. And then we have some options with our ascochyta blight and chocolate spot. We have PBA ambly as our best for chocolate spot, but there are a few other options there that are less susceptible, I suppose, than say your Faras and Fiestas. And then ascochyta blight, the ratings are there. But it's really important just to update and check those varietal ratings. They change every year. We've got really good funding from GRDC across the country, looking and trying to get good disease epidemics in controlled conditions so we can have robust ratings. We've revised the definitions, and so hopefully these ratings are going to get better and better over time as we get more and more data behind these varieties. So next slide, James. So how do the varieties look this year? These are some pictures here from Lake Linlithgow. I've gone through and taken the results from 29th of September. I didn't put together, I've just started going through pictures from yesterday now. PBA ambly is looking a lot worse for wear, but I'll tell you what, it's a lot better than say looking at Fiesta and Bendoc, which I think are almost gone. I wouldn't be running the header through them if it was me, but we need some data. So I'm sorry guys, you're gonna have to do it. But Fiesta and Bendoc are actually looking pretty similar. They're rated differently, so susceptible versus a moderately susceptible, but we're actually seeing they're very similar and sometimes swapping between the two depending on reps. Samira is a lot better than both Bendoc and Fiesta. And then ambly, if we look here at ambly, you can start to see the defoliation at the bottom of the stem, nowhere near as bad as Bendoc or Samira, but you can start to see those bottom leaves dropping off in that picture on the right-hand side of your screen. And then you can start to see those lesions going further and further up. The good thing about ambly is the lesions are smaller and there's less of them. But once it really does get going, and if it's got some disease pressure under it, we are seeing significant leaf area loss, and we've seen significant flower loss due to disease in PBA ambly. So I dare say it won't be a moderately resistant next year. Next slide, James. So this is just the data. This is an average across all our fungicide treatments. The fungicide by variety didn't come out as significant, and the scientist within me couldn't show those differences. But what we've got is an average across those varieties, but it really shows that ranking of varieties there, and what we've been talking about, what I just mentioned. The interesting thing is, in yesterday's data, I don't think that Bendoc Fiesta difference will be there. So as you get later in the season and higher disease pressure, that Bendoc is really falling over. So next slide, James. So the other trials that these trials we've been looking at that I'm going to go through are looking at fungicide actives. So on the left-hand side, similarly to the last graph, which I didn't talk about, percentage leaf area affected. And then we've got some different active ingredients along the bottom. It's like a baking recipe, I suppose you could say. Although I wouldn't be baking with these chemicals. So on the left-hand side, we've got Morava Star, which is a new Syngenta product. This is actually a group 7 and 12, an exciting new product that's coming out. I'm not going to try to pronounce those actives because I haven't started yet. Then we've got a complete control, which is multiple fungicides. Next, we've got tebuconazole and azoxystrobin or Varieties. We've got prosimidone, then we've got bixafen prothioconazole and carbendazim, all versus an untreated or no fungicide plot. So in this trial here, we were really trying to look at how those newer chemistries stack up against our cheaper prosimidones and carbendizoms that are Schedule 7, more dangerous to apply. They also need more regular applications. And so can we imply these other chemicals that have got more expensive active ingredients, then if we apply those active ingredients at the flowering time, can we get them through, or how far can we get them through the season? So I just saw a question come through. Which of these varieties is this across? This is actually an average across all those varieties there, but there is obviously a varietal effect on top of that. And I could present results on that, but we've just chosen to take the average at the moment, because that interaction between variety and that wasn't significant. It's basically showing the same in each one. So if it was in Bendoc, you'd just see those graphs would be just much higher, basically. But what we're showing there is those new active ingredients are quite good at holding back the disease, or just as good with this regime as what multiple, say, carbendazim applications are. And it'll be interesting to see how they hold up during the season, because some of those newer active ingredients are still holding up, whereas some of the older ones are falling over based on my visual assessment yesterday. So next slide, James, and I think it's back over to you.
Sure is. Thanks, Josh. So now we've got some varieties to work with. We've got fungicide options to work with. The question is, how do we start to put some of these things together? And that's what we're going to go through now. But we're looking at this because over the last few years, SFS has done plenty of trials, and we've shown, fairly consistently across a range of seasons that, generally speaking, an April sown bean will out-yield a May sown bean, and that our higher sowing rates have outperformed our lower sowing rates. There are exceptions to that, and it would be a great nut to crack if we could figure out why the exceptions are the exceptions. But as a general rule, we're seeing that increased biomass is increasing yield in our environment. But what we haven't done is looked at how the disease factor plays into that. So what we've done this year is we've crossed sowing rates with varieties, Amberley and Bendoc, with using fungicides or not to get some controls on what the fungicides are doing. And we've done that at Lake Bolac and Hamilton. So we're looking at combining all of the options that we have and seeing how they play out in their interactions. The first thing to show you is the big impact of the environment and the sowing rate. So this is Lake Bolac in orange on the bottom, and Hamilton, Lake Linlithgow on the top in blue. And on our x-axis is plants per square meter, going from 5 to 45 plants, and our y-axis is the percent of disease on October the 2nd. So what we can see here is that we have a consistent increase in disease with the increase in sowing rate, and we're seeing that it's a good deal more disease at Hamilton compared to Lake Bolac. So I think this is helpful for you all to see. When you think about the results that you're getting from other places, other environments, you can have this picture in your mind and think, is this Hamilton kind of results, or is this Lake Bolac kind of results? We've seen in the last couple of years, at least, that Lake Bolac has generally had less disease than Hamilton. Hamilton has just the conditions that are just rife for disease and pretty consistently. So the season is playing a big role. It's interesting to me that the slope of those lines is the same. So we're getting about 0.5% more disease for every plant that we add into the system, or that's 5% for every 10 plants. So it's, it's consistent. It's more how severe it is being set by the environment with a consistent effect of the sowing rate. Then the reason is, and we're sort of playing around with some humidity loggers. We're interested in seeing if we can monitor what's happening in the canopy and correlate that disease. The reason for that increase in sowing rate and increase in disease has to do with canopy humidity. So if you get a snapshot out of that data on July 28th, our red line there is the higher sowing rate, and the green and blue lines are the lower sowing rates. And you can see that the lower sowing rates is starting to dry down at 10 o'clock in the morning, but it's taking a whole extra hour for those higher sowing rates to start drying down. So they're staying wetter longer into the day, and that's favoring disease. And if you take a look more into spring on September 19th, with warmer temperatures, they're drying down starting at about the same time, 8 in the morning, but that higher sowing rate is still just staying more humid throughout the day. And so that effect is what's contributing to the greater level of disease with the higher sowing rates. But back to looking at combinations, what I've shown you so far is really averages of different treatments. So if we start to pull it apart, what else matters? When we pull apart whether we use fungicides or not, we see a really big difference. We see that we can actually keep the disease under control in all of our sowing rates. When we use regular fungicides, which I'll show you here, 4th of June, we used Aviator, 31st of August, Aviator again, and then Veritas the 15th of September. And this is scored a couple weeks after the Veritas. Bit of a longer window than we planned for, but just with weather, that's what happened between June and August sprays, and that's why there's a bit of disease in that bottom line. But that line doesn't increase. Fungicides are controlling disease at every sowing rate. Whereas if we don't control it, it's a linear line, it's a straight line up. More plants means more disease. So to me, this shows that our higher sowing rates don't necessarily need more fungicides at more times. That's being driven by the environment. The same number of applications, it's not like there are new opportunities, new times that we have to put fungicides out with a high sowing rate. It's more about if you don't get it out, if you miss it, if something goes wrong, the consequences will be much worse. So it's about making it a higher priority to get this under control rather than somewhere else. And then how does the variety play into this? What we can see is a really big impact with Amberley versus Bendoc. So again, plants per square meter on the bottom line and disease on the y-axis. Bendoc is increasing straight line up, pretty steep with plant density, but Amberley is tapering off and it's flattening out with those higher sowing rates. And so that's showing that the genetic resistance that Amberley is contributing is really playing a role at every sowing rate that we have there. And that's pretty exciting. That's showing that genetic resistance is sort of relieving the pressure on the system when we have those higher sowing rates. Lastly, to me, this raises the question, if Amberley is doing so much better, does that mean it doesn't need as many fungicides as, say, Bendoc? Can we get away with not spraying Amberley in some situations? And the results so far would suggest that it depends. So let me show you Hamilton versus Lake Bolac. This is Hamilton. On the bottom of the x-axis, we've got with no fungicides and then with regular applications. We've got an Amberley and a Bendoc line. And you can see that at Hamilton, Amberley is always better than Bendoc. Using fungicides is always better than not using fungicides, but the slope of those lines is the same. Basically, Amberley responds to fungicides as much as Bendoc does at Hamilton. But when you compare that to Lake Bolac, it's a big difference. Again, Amberley is always better than Bendoc. The fungicides are always making a difference, but Amberley is responding much less than Bendoc. Bendoc is getting a much bigger value out of that fungicide than Amberley is. The difference is if you look at the y-axis. At Lake Bolac, that y-axis only goes up to 40%, but our y-axis at Hamilton goes all the way up to 80%. So that's suggesting that Amberley's genetic resistance is helping in these situations, but there's a point when the disease pressure is so great that it will respond to fungicides as much as even Bendoc. But at Lake Bolac, where there was much less disease, Amberley is helping us a fair bit and not relying on fungicides as much as Bendoc was. So that's really interesting. It might suggest that, it definitely shows that Amberley is reducing the pressure on us to get our sprays out on time every time. But it's also showing that environment interaction, that it is potentially helping us much more. There's a point where disease is so great that even Amberley is needing fungicides as much as anything else. So let's put that together. In summary, we've seen that environment is key. Lake Bolac had less disease than Hamilton this year. It was the same last year. That might affect how we think about results when we look at other environments, when we look at certain years. We want to consider, what I'm hearing, is that relevant to me or not? I think Amberley might be in a similar sort of level of disease to Lake Bolac, certainly less than Hamilton. We also saw that environment had a greater impact than sowing rate. Sowing rate increased disease, severity of disease, but Hamilton was much worse for every sowing rate than Lake Bolac. And so that suggests that the environment is really calling the shots on what's happening with the disease, and the sowing rate is affecting how severe it is. We also saw that varietal resistance is underpinning all these decisions. So in some situations, and in most situations probably, you will still use fungicides on Amberlea. You probably, will never skip a fungicide with Amberlea that you would use with a Bendoc at Hamilton, but at Lake Bolac, maybe there would be some years where there's a difference. But the big thing is it's underpinning our decisions in the sense that it's taking away the stress. If you miss a timing, if it's not trafficable, if it's windy, Amberlea is holding up the fort a bit when you can't be there. And that is potentially very valuable in of itself. And lastly, we saw that the severity of uncontrolled disease increases with sowing rate, but not if the disease is kept out. So biomass is not creating new disease, that's the environment, but it is increasing the severity of that disease, increasing how bad it gets if it's not controlled. We can control it, especially with Amberlea. And I think all of this suggests, think of sowing rate really as being about biomass. You can get biomass either by increasing the sowing rate or by sowing early. And if you're going to be pushing beans, by doing that, by sowing early, by pushing the sowing rate up, you're probably not going to do that with Bendoc. The ones you're going to look to are Amberlea and Samira Ozara because they're going to hold up the fort a bit more when you're pushing the system. That's it from us. Thank you to the team. There's been lots of fungicides going out this year, so thank you to the team. Thanks to Agriculture Victoria. Josh has funded one of these trials. It's been great working with him. And most of this has been in collaboration with Southern Pulse Agronomy. So we're thankful to them and the grower host and their agronomist who had us. And I think it's time for some questions.
Yes, we've got a couple of questions in the chat box there. I think going back to Josh to start with, how cost effective is the complete brew of fungicides versus Miravis or Varieties?
Good question. In terms of cost effectiveness, these trials haven't gone through to yield, and I still say the proof is always in the pudding. In terms of, if I relate it back to chickpeas, I really hate to say this, the complete brew in a whole lot of chickpea economic analysis that I've done, although I would certainly never be recommending it to a grower, up to 7 and 8 fungicides is actually been the most profitable. It's actually significantly more profitable than applying 3 or 4 fungicides. And Jason's always said to me, Josh, really don't do the economic analysis, because he said you'll be so disheartened with what you do. He said every single time more fungicides is more profit. And it's probably good for the chemical companies. It's certainly not what we're condoning. How it fits here, it's going to be a very good question. I would argue that more applications of fungicide, you're damaging the crop every time you're going out there. There's a cost of application. So all of those factors will have to be put into it, and that's what we'll do in the economic analysis at the end of the year that we fully intend to do. The big other question out there is Miravis. How much, or Miravis Star, I should say it's Miravis Star, not Miravis. There's already Miravis registered in canola. This is Miravis Star, so it actually includes a second active ingredient. How much are Syngenta going to charge for that product? I can't say at this stage. It's under registration review. They're looking at having it registered for next season, by at least mid-season, and I dare say it's going to be priced at least Aviator. And after looking at the trials yesterday, they were really excited that it was looking better than Aviator, so let's hope they don't go too far out from that pricing. And the pricing of Varieties is continually changing with tebuconazole as well, going up in price.
This is one for James. What were the different sowing rates of Amberlea used to get the data in your last 3 graphic slides?
Yeah, I couldn't remember off the top of my head. They ranged, we went from big differences in plant densities from 5 to 45, and that, I think, from memory range from 30 kilos per hectare to 300 kilos per hectare. So we're not looking at realistic sowing rates for you. With these, we're creating data that's sort of showing more biomass, less biomass. Most of you will be in the 15 to 25 plants per square meter sort of range. I hear Hamilton people pushing 25 more often than Lake Bolac and Inverleigh people. But plenty of people are exceptions to that too. So I can look it up for you, and happy to get back the exact number for you. But something that really did matter this year is I have a photo on Twitter somewhere of like the different size of envelopes that we used to pack these trials. And for the same plant density, they were about twice the size of the other. And that was because Bendoc seed size, or it might have been Amberley, was much larger than the other one. And so that seed size really makes a difference. It's quite variable with pulses. And I would really suggest that you factor that into your decisions with sowing rates, because let's just say you weren't wanting a high sowing rate and you got some small seed, you might be a bit surprised by what comes out the other side. So seed size really makes a big difference, which is why I can't off the top of my head what the sowing rate was.
Do we have any data for the Western districts as to which bean varieties growers are sowing, Amberley versus Bendoc versus the older varieties? Have you had any conversations? It's been a bit hard because we haven't had our field days to talk to people, but
no, I couldn't say. I mean, my feeling is that Amberley is, got a lot of people's interest because of the disease rating. I hope the data was shown, makes people appropriately cautious with Amberley. I mean, I think Bendoc and Amberley has been a lot of conversations about because of the unique advantages they give. But Samira Zara are very consistently the most common varieties that I hear about, that most people who are interested in beans are using. Yeah.
Another question.
Samira's probably the biggest variety that's grown statewide, but Craig might have more data on what's grown in each area. I think he's got a pretty good scope, being more on that retail side, I think. I don't know if you want to comment, Craig.
Yeah, thanks, Josh. Yeah, fair to say that, big uptake of Bendoc last year. Obvious advantage there with the herbicide tolerance. And we've seen a lot of interest in Amberley, and I think as we get more and more information about how it's applicable in farming systems and the work that you're doing, Josh and James and Southern Farming Systems, is fantastic, proving the benefit of that variety. So expecting more orders to come through as a result of this.
So the next question I have is, once flowering has finished, do you think it's still worth applying more fungicide in a year like this with high pressure?
Josh, I'll let you take that one.
Definitely. What I can see out there now at the moment, you've got Bendocs that were a lot earlier podding. You've got Fiestas there. They're really struggling to fill their pods. They're totally defoliated, and they're going to have really small seeds because there's just not that green leaf area to be able to fill those pods properly. So it would actually be interesting to do some seed size analysis on some of these disease plots. It might actually be something to think about with those percent leaf sowing rate trials, James, as well. We might think about that later, because I can see that issue with that larger or greater leaf area affected. The other thing to think about is, what's your end use of your crop? If you're looking at marketing this crop for sale, pulses are sown based on visual inspection most of the time. And you've really got to think about what that seed looks like. And if you're getting seed staining for whatever reason, whether it be disease or weather at harvest, then you seriously have to consider minimizing that as much as possible.
So Josh, I guess the question raised for me is like, are you saying that there's a different way of thinking about fungicides during and after flowering? Like when we're in podding, are we thinking about preserving quality rather than other things like yield?
I think based on visual appearance this year, you can definitely see that the seeds are struggling to increase in size when you've got total defoliation. Now, how much leaf area affected do you need before you start impacting that grain size is a good question, and I don't think there's any data to show that anywhere that I've seen. In fact, there's very little data on a lot of this stuff that we talk about. It's all anecdotal. So I think you're doing both. You need to maintain enough leaf area to fill your pods, and that'll depend on the number of pods on the plant as well, I suppose, to see how much leaf area you need to fill that proportion of pods. So I think it's a mixture of both.
Are there more varieties coming along with resistance like Amberley in the breeding lines?
Don't know, but I'm talking to the breeder tomorrow, so I'll ask.
There is a lot of lines coming. Amberley was crossed into the program as soon as it was identified that as resistance. Clearly, they'll have an objective of higher chocolate spot resistance because it's going to be a big uptake. All the breeding programs are also moving to a large proportion of IMI tolerance. So you would expect to see something with IMI tolerance and at least Amberley resistance in terms of chocolate spot coming out in the near future.
Are there any winter favour beans that have some vernalisation to maybe allow for earlier time of sowing?
I think I have the same answer, John. I'll ask the breeder tomorrow.
And we've got one more there. Has there been any fungicide resistance detected in favour beans?
Good question. At this stage, what I'll put is in all of pulses, there's only been one detection of fungicide resistance in all of pulses. Interestingly enough, that was next to a grapevine or a vineyard in chickpeas and to botrytis. So using carbendazim. So it was resistance in chickpeas to carbendazim. And they were spraying, and we've all seen, well, I hope everyone's seen grapevines and how good our growers are in broadacre cropping compared to horticulture and the application methods that fungicides are used for. And that's where that resistance was found. So I wouldn't suspect it in broadacre cropping, but we're certainly monitoring with the CCDM. I took samples from a Lake Linlithgow site the other day, and all of those will go off to the CCDM once I've talked to them. And hopefully a lot of those will get tested. So it's something we're actively monitoring for, but there hasn't been any fungicide resistance at this stage. But yeah, based on what we're seeing in chickpeas, I'd certainly be wary of it. And that's why we're certainly actively promoting that rotation of chemical groups and fungicide actives.
Another question here. Are there any PGRs for favours?
I've heard, I mean, from what I've heard from like Jason and what other people have done, they tried this, they really worked on it, and it just didn't really come to anything. So if you've got really tall beans, windrowing at the right time might be your best bet. I assume that's why someone's asking. I have read a paper, you know, that they applied a product and it helped with, you know, it helped with the harvest index, but like that's like experimental stuff. So there's not really anything that's reliable at the moment.